I'm Dr. David Outomuro, a teaching assistant professor at the University of Pittsburgh. For this biography interview, I'm talking today with Dr. Maydianne Andrade, who is a professor at the University of Toronto, Scarborough. Welcome, Maydianne.
Thank you so much. I'm happy to be here.
Maydianne is the senior author in a paper called Precopulatory Choices Drive Postcopulatory Decisions, Mechanisms of Female Control Shift Across Different Life Stages, published in the journal BMC Ecology and Evolution. Today, we would like to chat about the hypothesis for that paper and some of the graphs in Figure 1. So Maydianne, could you please orient us to this graph we've been working on?
Sure. You picked a complicated one, which makes it more fun to talk about. What you see in this figure is the result of two sets of experiments in which we exposed females that we'd raised in the lab to a male for the very first time to see whether or not they'd mate and what that mating looked like. So the very top panel is the first mating of a female. So before this, she's never encountered a male. The second panel on the bottom is when we expose the female to a new male after she's already mated once. And we had a series of questions about this experiment. First of all, we wanted to know whether or not adult females actually would accept any male who tried to court them at the beginning when they first mate. And we predicted that because in nature, adult females actually have a relatively high risk of not getting a mate at all. So we have lots of field work that shows that at the end of a mating season, you have some females who, for whatever reason, they're in a bad location, their sex pheromone hasn't traveled very far, or maybe they're a low quality female. They don't get a male coming to court them at all. That is really a high cost for females in nature because they might die over winter and actually never reproduce. So we predicted that when an adult female encounters a male for the first time, she would mate what we call indiscriminately, without any choosiness or selectivity about that mate. We then said, well, if that happens, it might be that the benefits of being choosy come in the second mating. And that is that females may do things to allow a second mate who they allow to mate, to allow a second male who they allowed to mate, to father more of their offspring in some situations, but not others. So there's a lot of pieces here. One is initial female choice, which you see on the top panel. The second is what females do with the second opportunity to mate, given what they did the first time. And then the other thing is that we use the fact that females can mate either as adults or as subadults to probe whether or not that risk of not mating is what is making the difference between their behavior in the first opportunity. Subadult females, we thought, they're not even sexually mature yet. And in this species, they can mate, but that means that you don't have as high a risk of, let's say, going through the whole season without mating if they're already encountering a male as a subadult. And so in this figure, then, what we show is a variety of things starting from the beginning, pre-copulatory effects, all the way through to mechanisms that happen after an individual is copulated that might affect whether a subsequent mate can actually father more offspring or would be blocked from fathering offspring. So first of all, we have pre-copulatory cannibalism in these species, in the species of brown widows. Sometimes females will kill males as they approach them during courtship. And as you can see, there's really no difference between adult and subadult females and whether they do that. There's whether or not the female mates at all. And in what we found against our prediction was that both adults and subadults tend to be fairly what we call indiscriminate. 80% of them mated with a male regardless of what that male's phenotype was. He could have been large, he could have been small, it didn't matter. And the third one is a number of copulations and the number of plugs on the fourth one that the female has left after that mating. And the number of copulations and plugs together will determine whether or not the first male will fertilize all the eggs or whether a second male will have a chance to fertilize eggs because of the fact that the female can plug the male can plug the female's genitalia and make it impossible for next male to mate, but only if the female allows him to mate twice and to place two plugs. So what you can see in the graph in general is that subadult females and adults are equally indiscriminate, but adults leave that opportunity open for a subsequent male to fertilize eggs much more frequently than subadult females do. And so in the second graph, once they've mated once, the adult female is much more likely to remate and give that second male the chance for paternity.
Thank you. That was very interesting. Can you explain why you use these model species? What makes them special?
This particular species, the brown widow spider, Latrodectus geometricus, is one of 23 species of what we call widow spiders. So you've likely heard of a black widow, the southern widow, the western widow, but it turns out there's 23 species worldwide. And so this species is unique not only because of this unusual subadult mating, which we found in a few other species in the genus, but also because we feel that if we build out our understanding of the entire genus of 23 species, we will have what you call a model clade. So instead of just a single species where we can ask questions about what's happening within this species, we can now start to ask comparative questions that allows us to get at broader evolutionary hypotheses. If we have 3 species that show this form of mating and 6 of them that don't, can we make predictions about what causes the evolutionary cascade that leads to one set of traits versus another? So that's why we did this work on this species.
So I think you already partially answered my next question, but tell me a little bit more about, for example, where that data come from, or for example, within your research program.
Thanks for that question. I always tell people that when I started working on widow spiders, so genus Latrodectus, I would go and give talks. So as a master's student, for example, I started in my master's, so I've been working on Latrodectus for a long time. And the most common answer to questions at the end of my talk was, I don't know. I don't know. I don't know. Here's what we think. Here's what we, infer. Here's the little bit of data I have. And so basically I've spent, more than 2 decades building up a body of knowledge to get to the point where we think that this group of animals could be useful for answering broader evolutionary questions. So really it's been from very little knowledge to gradually putting the pieces together.
Oh, that's great. Yeah. Could you describe if you did any follow-up experiments or potential follow-up experiments that you're planning after these results?
So we have been working on this question of subadult mating for quite some time, and we now have 3 different species that we've demonstrated that this occurs within this genus. And more generally, for the first, I would say, maybe 15, almost 20 years of my career, we focused primarily on decision making by males. And there's a variety of reasons for that. One is in these species, males have this like spectacular somersault behavior where they kind of throw themselves onto the female's jaws. It's very visible. It's very, obvious that it's something puzzling. Females live much longer than males. So males live a maximum of a month to 3 months, even without cannibalism. Females can live for 2 years or more. And so it was challenging to get data on females and to ask questions about female biology and decision making. But with the buildup of all this information we have, we now realize that we can ask questions that are tractable with females. So, for example, we've now shown that females can detect the social environment around them. They can smell males at a distance. And they use that information to tune what you see in this paper. And so if they smell lots of males, they are choosy on their first mating. But in this experiment, for example, where we reared them in isolation and they couldn't smell males, it's mostly airborne pheromones, they are not choosy because to them, they're in an environment with very few males available. So we've started to get a really nice feel for how female strategies are intersecting with male strategies in the species.
Thank you. That's very, very, very fascinating. I'm very curious to know what kinds of scientific questions interest you the most.
I think for me, my interest in biology started with an interest in behavior. And I'm particularly fascinated by mating behavior, although I have to admit that part of that, I found very much so, it depends on your professors. So as an undergraduate, I had these outstanding animal behavior professors, and I was in a department that had a lot of animal behavior. I had a professor in intro biology who I still remember very distinctly. Every lecture, and back then we didn't have slides. They were writing on an overhead if we were lucky, right? So every lecture would start with a story about research. And he told it like a story and connected it to the theory. And really that's what drives me is these questions that arise from watching things in the natural world, asking how they fit into theory, and then testing whether or not we have it right. So I sort of say to people, I'm kind of a control freak. I would love it if the whole world was one big equation that predicted everything. I know it won't be, but the closer I get to that, the happier I am.
Thank you. Could you share with us when and how did you know that you wanted to be a scientist?
Yeah, I think, again, I'll come back to being a control freak. I really like the idea of understanding things, and I always have. I had a really, again, I come back to the importance of teaching as well. I had a wonderful teacher in grade six, and I'd always liked science because it has rules. I like rules, right? And I like understanding rules. And so, but then I remember in grade six, one day it was raining outside, and we're sitting in this classroom with windows. And he said, without getting up from your seat and without touching anything, I want you to prove to me that the water's coming down from the sky instead of up from the ground. And I remember thinking, oh, that's easy. And then we started talking, and I'm like, that's not easy. Like, it took us the whole class of walking through hypotheses and predictions and how we could test it without getting up. And I was hooked by that. And then when I got to first year, my professor, again, this professor telling these great stories about research. But I think maybe because I'd never had a woman professor. I'd never had a Black professor. I had like one professor who was racialized, which is what we call people of color in Canada. It never occurred to me that I could be a professor until about halfway through that intro class. And I actually distinctly remember the moment when he was telling one of these stories, and like the hairs on the back of my neck actually stood up, and I thought, oh, I could do what he's doing. Like, this is a job. People do this for a job. And my mind was blown, because before that I'd said, oh, I'm going to be a doctor, because that's kind of what everyone who likes biology says. So it was really having my eyes open through those stories about research. Yeah.
Thank you very much. Could you tell us a little bit about the paths that led you to your current job?
Yeah. So I would say great mentors and great mentors, great instructors, and serendipity and luck, really. So in the end, that same first year professor who told those great stories and inspired me to want to become a professor was my second employer. I was in a cooperative education program, and he had a position in his lab, and he recruited me for that position, and that really got me started on research as an assistant. And then when I decided that I wanted to stay in research, he was the one I went to say, who should I apply to for graduate school? And then I'll try to say this without crying. He showed me the letter of recommendation he wrote for me. Yeah. And, one of the things it said is her biggest flaw is she doesn't know how good she could be. And I'll never forget it, right? I don't think at the time people know so much about so-called imposter syndrome, or the effect of being like I was the only Black woman. I was the only Black person in my entire cohort of 250 students at my undergrad institution. I can't even describe how important that was. And this was a white man of a certain age who just took it upon himself to be, such an important mentor to me. Yeah. And then the serendipity part was I actually had in my final year, written a research proposal about sexual cannibalism. There was a big debate in the literature at the time, that's really going to date me, about sexual cannibalism and the possibility that males could sometimes benefit from being cannibalized. So I wrote a research proposal about it. And when I wrote to my potential master's supervisor, I sent that as an example of my writing. And then when I got to his lab, I thought I was going to work on what he was doing. And he said, oh, no, you're going to do that cannibalism thing. And that was the start of my career, and I'm still doing it, decades later. So that's what I say, kind of luck, serendipity, and great mentorship.
So we all know that university professors are very, very busy. So what is a typical day for you?
A typical day for me now is busy every single minute of the day. I think it's hard to describe the extent to which, if someone stops you in the hall to talk, your whole, there goes the time I had to write this one document kind of thing. So it's rich though, right? So I walk in and my PhD student has a question or has some great new result that they're excited about. We might have a lab meeting. Unfortunately, I have a whole bunch of other meetings, but a lot of them are about good things, about, mentorship programs, about some of my service groups like the Canadian Black Scientists Network, where we're applying for grants to support various kinds of things. Earlier today, I gave a keynote talk, not on biology, but on equity, actually, and how the importance of being able to talk to people in a realistic and grounded way about why you think fairness is important in our context.
Thank you so much. So given that you are so busy, how do you balance work with any other interests in your life?
Yeah. So I have two kids, and right now one of them is 22. She's actually went away to do her undergrad, came back to do a master's in Toronto. I have a son who's 17 who's just been accepted to university, so we'll be heading off next year. And when they were little, I didn't balance it very well. I mean, my husband's also an academic. We worked and we took care of the kids. We worked and we took care of the kids, right? And now I'm finding as they're older, I'm able to engage a bit more with some of these other things. Like undergrads always want you to come to something that starts at 5 p.m. In my old, 10 years ago, I couldn't do that. Now I try to do those things when I can. For me, one of the really hard things to balance, though, is that I am engaged in a lot of what I call systems change work, trying to build in more supports that currently don't exist, trying to write the unwritten rules, whatever you want to say, for people like me who are maybe first in family at university and for whom it was all like, what is going on here? And I don't have as much time for individual mentorship as I might like. So in my lab, I have 5, 6 undergraduates. Some of them are doing research. I have my graduate students. I had a postdoc. And then I have my family, I have my friends. I can't do it all. So I've had to accept that this service work takes the place of some of the individual mentorship. And so I make myself available in these like 5 p.m. group things where I can talk to people about these things. So that's how I try to balance it. Oh, and then of course the gym. I really enjoy weightlifting, although I don't go as often as I should.
So can you share with us what is like being a Black scientist that is the first generation in your family to attend grad school?
That has been interesting. Being first in family is kind of wild. And I say to people, you either need your family to be supportive or you need to have a found family who's supportive, because it's an unusual life to begin with, to be in grad school. And so I remember my parents, my friends, everyone would say, "Oh, are you coming home for the summer?" It's like, no, that's when I get all my research done. I'm not coming. I'm not a teacher. I'm a researcher, right? So there was just a lot of things I had to explain along the way, and they were very receptive to that. I study spiders. So there was a period there when I was an undergrad where there was always bugs in dead containers in my mom's freezer. She learned to tolerate that. But it has been, it's lonely at times. Like some years ago, someone wrote an article about teaching evolution, which is what I do. And they said, "Oh, we're going to do these interviews with people who teach evolutionary biology, and you'll all be anonymous, but we'll have these comments in the article." And when this article came out, it said, Jane Doe, whatever they called me, a Black woman teaching evolution at university. And I said, "You have identified me as an individual in Canada." That's how few people there are, Black women in biology to begin with, and in my field in evolutionary biology. So, there's a lot of people wanting you to do things for them based on your identity. But when you have good friends and colleagues and mentors, you learn how to filter those things out. And really, I've had wonderful colleagues. And coming in, to be honest, I was lucky. My husband and I got jobs at the same time in the same institution, made a big difference. Yeah.
Thank you so much. My last question for you today is that if you could please tell us about a moment when you felt like you really, really belong in the field of science.
Yeah, I think this is going to sound silly, but when I was doing my master's degree, so it's when I first started working on Black widows. First of all, I was afraid of spiders to start with. That's a whole nother story. But after I got to know them better, I got over that, and I was just so interested in the questions about cannibalism. But no one else was working on spiders. So I learned about how to raise them, how to do the experiments by going to the zoo where they had a rearing facility, and then just reading the papers. And just so your people in the audience recognize this, when I was a graduate student, you would get a paper for someone by sending them a little card in the mail, in snail mail. And then they would mail it back to you. So you'd wait for like a week or 2 weeks, and then you get this paper and you'd be all excited and read it. So I'd only ever heard about these behaviors. I'd never seen them. I'd never done the dissections. I just did it by looking at the papers. So I just remember the males of this species I worked on from Australia does this somersault I described, where the male twists his body into the female's fangs. It turns out the courtship lasts for 8 hours, which I didn't really clock when I read the paper. So I was sitting, I remember just having all my things around me. I'd set up the stuff. I had to work overnight by myself because they're nocturnal, and I didn't have any way to turn the light cycle around. And I remember being there at like 2 a.m. and they mated. And it looked exactly how it was described in the paper. And I just ran around like yelling. I found a custodian and I said, you have to come look at this, look at this thing. And it just made me realize like the literature is real. These things that I've read about and these ideas I had about, the experiments I wanted to do, it was all real. And it just really grounded me in the joy of being a scientist and that it was accessible to me.
Thank you very much, Maydianne. It's been wonderful chatting with you today.
Oh, really nice talking to you too. Thank you. I appreciate this. I actually love chatting about this stuff.
If you want to learn more about this work, you can read Precopulatory Choices Drive Postcopulatory Decisions, Mechanisms of Female Control Shift Across Different Life Stages, which was published in BMC Ecology and Evolution in 2023. We've included a link to it below this video.